So
Okay.
as I welcome all the participants and this is a good evening, very good evening to all. And as you all know, today's speaker is Dr. Tanmay Majumdar sir. He's a staff scientist at National Institute of Immunology. You all know this is DBT, Department of Biotechnology run institute. Uh, you can say first or second DBT sponsored institute. And sir is head, Dr. Tanmay Majumdar is head of vaccine development unit or center. And this is center of excellence also. And he is also a member of editorial board of so many, you can say journal, including the scientific report and other journal also. He's in editorial board and editor also. And as you all know that I don't discuss more about he has long publication and other things. So with this brief introduction, I welcome Dr. Tanmay Majumdar sir in this science dialogue series. And most important thing is this is free webinar series, and he is ready to deliver lecture. He has accepted my invitation. That is also a very important and very, you can say, grateful thing, because people are expecting so many things from different platform. So all those who are delivering lecture, they are very eminent scientists in his or her domain, in different domains of the bioscience. With this, I have first time from NII, National Institute of Immunology, scientists are delivering lecture. So I heartily welcome, and we are grateful to you. Now, platform is yours, sir.
Yeah. Thank you, Dr. Gopalji. It is my great pleasure to present our work in this August gathering. I believe that many of the participants are undergrad and grad and postgrad students.
Yes, and faculties are also.
So based on that, I have made my slide so it can be understandable for global audience. So here I am sharing my slide. So I think you can see my PPT, right? Can you see my PPT?
Yes, yes, yes. It's
Okay. So this is National Institute of Immunology. It's a fantastic campus, perhaps the one of the best uh, landscape we have across the India, whatever the institutes are there. So this is the main building. Uh, so this is the main building, okay, and this is the reception area. And we, the faculty, whoever in NII, they generally stay inside the campus. So this is the residential bungalow where the faculty families they live. This is the area of playground and other parking area. This is the another area where kids do play. It's known as the rock garden. So it's very beautiful place. It's adjacent to JNU and Delhi IIT. In between it is there. And JNU has the natural landscape of mountainery, and similar mountaineering landscape we have at NII, and we maintain that landscape. And maintaining that landscape, this building was made. It's a fantastic infrastructure what we have. So NII is there is government of India has one ministry, that is the Ministry of Science and Technology, and under Ministry of Science and Technology there is a one forum is there, that is BRIC, Biotechnology Research Innovation Council. This is under DBT. See, this Biotechnology Research Innovation Council has 13 institutes across the India. So these are the different institute like Center for DNA Fingerprinting and Diagnostic Institute, Institute of Bioresource and Development Institute, Institute of Life Science. This is at Hyderabad. This is at Manipur. This is Institute of Life Sciences at Bhubaneswar. INSTEM is at Bangalore. Navi is at Hyderabad. NBRC, National Brain Research Center at Delhi. National Center for Cell Science at Pune. National Institute of Agriculture and Food Biotechnology is at Chandigarh. National Institute of Biomedical Genomics at Kollani, Kolkata. This is NII. National Institute of Plant Genomic Research. This is adjacent to NII. And this is Rajiv Gandhi Center for Biotechnology. It is at Thiruvananthapuram. And this is the Translational Health Science Technology Institute. This is also in Delhi NCR in Faridabad. So these are the institutes which are under DBT, and whoever here the undergrad students, they have the option, they can choose to do their master degree in those institutes. Many of the institutes like THSTI, RTCB, NBRC, they have their master program in STEM. So if you want to, if you do not want to go in university master program, postgraduate program, you can join here in this postgraduate program. This is also under Regional Center for Biotechnology. This is a university, deemed university under Government of India, where under which there are multiple courses are there, master degree program are there in THSTI. Like there is a program called Human Translational Research. So based on this subject, you can do the master degree and followed by you can explore your career. So keep an eye on all these websites, and you can find out that they do have the master program along with whoever done the master degree. All these institutes have their own PhD program. So even if you have the CSR DBT fellowship or not, you can apply in those PhD program, and you can continue, you can register yourself as a graduate student. So this is the National Institute of Immunology, which has been made in 1981, and then DBT was not formed, so it was under DST. So DST, but when DBT was made, then DBT, this institute was transformed from DST to DBT, and this is the one of the autonomous institute under DBT. So the mission of this institute is like that: is promote, guide, and coordinate the research of high caliber basic and applied immunology, and to promote effective linkage on the continuing basis between various scientific and research agencies, laboratories, other organizations like companies, entrepreneurship, working in century in the field of immunology, vaccine development, and related areas. To organize postgraduate courses, workshops, seminars, symposium, training program of the specialized nature in the field of immunology, vaccine development, and related areas are done here. So goals are that to provide national and international leadership in immunological sciences, providing quality healthcare solution to countries' major medical problem, expansion of immunology science by incorporating tools, methods of chemistry, physics, and mathematics, and playing a leading role in creation of immune engineering, like CAR T cells and all. So research validation, we have made the anti-leprosy vaccine, which is in market. We have made the anti-cancer immunotherapeutic dendritic cell-based vaccine, which is also in phase 3 trial. Product development, cell intellectual property rights cell provide vital support in efficient transfer of research to industry. So present status, emphasis of the research at NII, design new and more potent vaccine against the infectious diseases, considered as a premier center in the modern biology, basic immunology of host pathogen interactions, extensively published good our research work in reasonably good journals, and internationally acclaimed research institute has been considered. That's why this NII. Now let's come to the science. So as this is the Institute of Immunology, and myself, my training is under immunology subject. So we talk about that. What is immunology? Immunology, the subject which talk about that our immune system. And immune system is divided mainly in two categories. One is innate immune system, another one is the adaptive immune system. So innate immune system, these are composed of, one is the physical barrier, another one is the chemical barrier, another one is the innate response. Like there are different antigen presenting cells are there which play an important role. On the other hand, that adaptive immune response are divided into two arms. One is the cytotoxic T cells, CD8 based T cells, cytotoxic T cells, and CD4 T cells, which are cell mediated immune response. Another was the B cell mediated immune response, which is known as humoral response, which produces the antibody. So these are the two different arms. One is innate immune system, another one is the adaptive immune system. So immune and adaptive immune system, these are the different cells which are part of the innate immune system. One is monocyte, macrophage, mast cell, dendritic cell, NK cells. These are the different cells which are the part of innate immune cells. Along with these are the cells which are part of adaptive immune cells like T cells and B cells. So B cells can produce different types of antibodies, and there are 5 types of antibodies we have. One is IgG, IgM, IgA, IgD, IgE, based on the where you are immunizing the vaccine candidate, and these are the different organs. So which are playing important role to develop our immune system. Main was the bone marrow. Bone marrow is the place where from the B cells and T cells are formed, and these T cells generally goes to the thymus where it get mature, get education. After education, it been distributed to the different peripheral organ like in spleen, lymph node, tonsils, etc., etc. So the innate immunity, that suppose this is a cell which is maybe macrophage or dendritic cells, it express lot of pathogen recognition receptors. So pathogen recognition receptors, like these are the pathogen recognition receptors which are helping to recognize the pathogen like Salmonella, Vibrio, SARS-CoV-2, all these bacteria, virus or parasite like Listeria, Plasmodium. These are the different pathogens are there. Like one is category is virus, another category is bacteria, another category is the parasite, another category is the fungus. So these they have specific pathogen associated molecular pattern, and this pathogen associated molecular pattern is being recognized by different pathogen recognition receptors which are present in our macrophages or dendritic cells or any other monocytes. And these pathogen recognition receptors are like TLR1, TLR2, 4, TLR5, TLR6, TLR10, which present at the membrane. Some of them are present at the cytoplasm also. In the endosome also, TLR3, TLR7, TLR9. There is another pathogen recognition receptor which is known as STING, AIM2. These are the different pathogen recognition receptors which recognizes different pathogen associated molecular pattern. So like here you can see that TLR1 can recognize the peptidoglycan, TLR4 can recognize the LPS, TLR3 can recognize the RNA. So these are the different molecules which are coming from the different pathogens, and these pathogens are being recognized by these pathogen recognition receptors, either TLR molecule, NOD molecule, NLRP3 molecule, dectin, RIGI, MD5, etc., etc. And this pathogen recognition receptor, when it binds with the pathogen associated molecular pattern, it gives a downstream signal which is MRDA dependent or independent. In independent TRIF is involved, and that helps to activate the interferon pathway, interferon induction pathway, where different interferons are released. So as I have told you again, I am repeating, this pathogen recognition receptor, you have to remember these TLRs which TLR is recognizing the which pathogen associated molecular pattern, and when these TLRs is not only in plasma membrane, they present in the cytoplasm like TLR3, TLR7, 9, TLR3, TLR7, 9 present in the endosome also, and STING also it says another pathogen recognition receptor which present in the cytoplasm that recognizes the DNA of the pathogens. So when the these are the innate immunity which activates further after releasing the different cytokine. It helps to activate the T cells. Now, how the T cell develops? So first, the T cell is coming from the hematopoietic stem cell. This is the from the bone marrow, and from the bone marrow, it differentiated into progenitor T cells, and this progenitor T cells actually it goes to the nucleus and pre T cells, and that is differentiated into first is double negative, then further it goes to the double positive, and then it differentiated into CD4 or CD8. So here you can see that this the thymic stromal progenitor cells, it enters in the thymus. This is the thymic cortex region. Suppose this is the whole organ, this is the thymus. So this is the thymic cortex region, and this is the thymic medulla region. So in thymic cortex region, first there is a double negative, and double negative from there it produces that double positive cells. These are the double positive cells, and from double positive cells, the CD4 and CD8 is being differentiated. And in the process of development of the T cells, different signaling factor like Notch factor, beta catenin also plays important role, beta selectin. These are the different factors which plays in T cell education. So further, when T cells become educated, then T cells, what happened? It expresses the TCR, like TCR which can bind. You can say this is the macrophage, and this macrophage has major histocompatibility complex. This is MHC2, and this is either MHC1. When MHC2 is there, then T helper cells are coming, which is CD4 positive, CD4 T helper cells. When MHC1 is there, then cytotoxic T cells, which is CD8 positive, they are coming and they bind, and it gives a release of different cytokines. And T cells are being differentiated into effector memory cells as well as effector T cells as well as memory T cells, and some of the T cells also get exhausted. That is the exhausted T cells we call. Effector T cells can differentiate into different T cells like Th1, Th7. This is the Th1 which has the Tbet, which helps in release of interferon gamma. Th2 which has the transcription factor GATA3 or STAT6, which helps in Th2's differentiation. The ROR gamma T is another transcription factor which helps in IL-17 release that differentiates into Th17. Also, there is the anti-inflammatory T cell, that is the Treg, which has the FOXP3 CD25 that helps in maintaining the anti-inflammation. So, based on the different cytokines like TGF beta helps in Treg, IL-6, IL-23, TGF beta helps in IL-17, IL-4 helps in Th2, IL-12 helps in Th1, and there is a T follicular helper cell that helps in memory response, ICOS, IL-6, IL-21 that helps in T follicular cell differentiation. And when these T cells also activate the B cells, and these B cells, when there is a clonal expansion, and this cause B cells either differentiated into memory B cell as well as the plasma B cell. Plasma B cells are the B cells which release the antibody to neutralize the pathogen. So this is the common overall concept what I have given, that how T cells are formed. From the B cells actually formed in bone marrow cells. T cells, precursor of T cells can form in bone marrow, but it goes to the thymus, and there its education happens. So after proper education, the T cells differentiated into either T helper or T cytotoxic T cells. So now question is that you remember, for the students, what I would like to let you know that before COVID, and the whatever the vaccines are available, it was given to the kids from the baby who was born from 0 to 12. So in between that age group, the people used to student kids are used to get the all the vaccine. Why not at the adult stage? Why not at the older stage? Except the flu vaccine, which are given in USA and other Western countries, all the vaccines, whatever available before COVID, all are given at the younger ages, not the older ages. The reason is that because thymus is the critical organ which helps in T cell education, T cell differentiation, clonal expansion, and thymus is getting shrinkage at the increasing age. So when at the age of 50 or 60, the thymus is almost shrinkage, and because of that, as they do not have the thymus, so vaccine memory response, vaccine efficiency goes down. That's why to get the efficient vaccine response, all the vaccines were candidate whatever available across the globe, it was given to the kids. And after COVID, when COVID vaccine has come, you have seen that those COVID vaccines are not equally effective to all the humans. In your family, you have seen that even some of them who has the COVID vaccine, in spite of that, they have the further infection or further symptom. That is because of the T cell memory response was not sufficient, because thymus was not active that much what which it was in at the younger age. So because of the incapability of the thymus, the vaccine response goes down in course of your age. So it is always remembered that thymus is the critical organ which is required for successful vaccine efficiency. So now how the T cells work, how the T cell is, so there are three signal is there, signal 1, signal 2 and signal 3. These are the three critical signal which are important for T cell activation, clonal expansion and differentiation, because this one signal is that is MSC. MSC is present in that the antigen presenting cell like macrophage, and T cells has the TCR. So TCR MSC interaction is one of the critical step which is essential for T cell activation. Second important molecule is that co-stimulatory molecule. These co-stimulatory molecule are also important. Without that, this even there is interaction of MSC and T cell TCR, but there would be no activation. Third is the cytokine cues. These are the different cytokines which are released by these antigen presenting cells which are expressing the MSC, and these cytokines actually helping the T cells in differentiating at the different T cell subsets, like IL-12 helps in Th1, IL-2 now people are saying it is helping in Treg, IL-15, IL-21 these are helping in Th15 and T cell 15, and IL-22 are also helping Th17. So these are the different cytokines which are required for T cell differentiation, along with signal 1 which is TCR MSC interaction. Co-stimulatory molecule are also critical for T cell activation. So these are the three signals you have to remember to how the antigen is being present and being that is recognized by the T cell for T cell activation, clonal expansion and differentiation. So now question is that these T cells are very much important for our immune system, but how does T cells are being regulated by our microbiota? Microbiota is the population of different good and bad bacteria. Not only bacteria, fungus are also there, bacteriophages are there. These are generally present in our gut, in our lung, in our skin, in our different mucosal organs. So wherever the mucosal organs are there, this gut microbiota is there, and this gut microbiota is playing a critical role for successful of vaccine or successfulness of any medicine or drug. So this is the lumen, and this lumen, this is the mucus. This gray area is the mucus, and this from the mucus, the microbiota gets the nutrients for their survivals, and these epithelial cells sometimes they modify and they function in different way, like modified goblets epithelial cell is goblet cell. This is also kind of epithelial cell that helps in providing the nutrients to the microbiota. M cells it act as an antigen painting cell, like it can express the different TLR which maintain the homeostasis inside our body. And third is the Paneth cell, which release different antibacterial peptide like REC3, beta, gamma, mucin, defensin, etc., which helps to neutralize the pathogen. So when this TLR is being recognized by the pathogen associated molecular pattern of the bacteria, it gives a downstream signal. It releases, that's why I told you, the signal 3, which releases different cytokine like TSLP, thymic stromal lymphoprotein, TGF beta, which helps with dendritic cell. This is the lamina propria, where dendritic cells are there, and that release the retinoic acid, and that goes to the mesenteric lymph nodes. So lymph nodes are there where T cells are present, and with the help of between dendritic cells or macrophages, these T cells are differentiated into Th2 or Treg that maintain our tolerance. Tolerance means even these are the microbiota is our foreign material. It is not our own thing, but still it is called. But in presence of this microbiota, our body is maintaining the homeostasis condition, and that is known as tolerance. And this tolerance, so this is the microbiota. You can say that a galaxy contains 100 billion stars. Remember that a galaxy contains 100 billion stars, but our gut contains 100 times more than that bacteria. Means 100 billion stars are present in only in gut. So if you compare the genomic material, where we have the 23,000 genes, but human microbiome has more than 1 million genes. So its body contains 100 trillion non-human cells. Like the non-human cell means microbiota, that 100 trillion, which is 10 times more than our total body cells. 99% of our DNA is coming from microbiota, not from the human cells. So that's why microbiota is known as a second brain of the body. So you remember that this microbiota is playing a critical role since the baby born till the person die. So this microbiota is important to maintain the efficacy of our immune system. So infant's microbiome is determined by the when the baby born. You remember that my baby can born in two ways. One so maternal offspring exchange is important. So it becomes similar to the adult microbiome by the 3 years of age. The total microbiome composition of infant mostly dominated by the Firmicutes, Pteridicutes, Proteobacteria, Bacteroides, Fusobacteria, etc. So we have the baby can deliver can born in two different ways. One is the vaginal delivery, another one the cesarean delivery. So cesarean delivery always not recommended because they do not get the different good bacteria which are provided when there is a delivery through vagina. That's why lactobacillus, pivobacteria, these are the important good bacteria which are present. But when the cesarean delivery is there, these bacteria are missing. So, but breastfeed milk infants also augmented with bifidobacterium, streptococcus, lactobacillus. That's why breastfeed male infants have the more microbiome diversity, and that helps to develop good immune system. Whereas bottled milk fed infants has the reach of aerobic bacteria, lower frequency of bifidobacteria or other good bacteria. So diet and microbiome has the important interaction to develop the immune system. And within the first couple of centuries of the earth, the microbiome was saved by whatever the hunters and gatherers could arrange by their community. These may have included berries, nuts, seeds, lean meats, etc. So these things, so these are the different microbiota, how it regulates the different T cells. Like Bacteroides is a gram-negative bacteria, Clostridia, segmentus filamentous bacteria. These Tregs are differentiated by these are the Bacteroides or Clostridia. These are the good bacteria. Th17 is differentiated by segmented filamentous bacteria. So this causes the inflammation. So here you can see that there are other nutrients are there which also is being controlled by this microbiota, like Bacteroides, Prevetola are helps in release of short chain fatty acid, butyrate, acetate, vitamin A, which maintain the balance between Treg and Th17 inflammation, anti-inflammation versus inflammation, and that's why body maintain the homeostasis. Imbalance of vitamin A causes the enrichment of Th17 cell and small intestine, which associated with the lot of inflammatory bowel disease, where segmented filamentous bacteria goes high. Thus microbiota frequency are the indispensable factor for efficacious immunization, and you can remember that these are the different bacteria which helps in carbohydrate digestion and all. So now here this is the very important picture. You remember that since the baby is the fetus, so from since it borns, this is the infant newborn baby, was followed by 2-3 years, 6 to 8 years, and adults. The vaccine efficacy and immune system development is very much dependent upon the microbiota. So the placenta health, mode of delivery, either vaginal delivery or cesarean section, breastfeeding or bottled milk fed baby. Based on these are the different maternal factors which are critical to maintain or develop the high diversified microbiome inside the infants, and that helps to develop the good immune system. Not only this, there are other factors, environmental factors are there, like food, pathogen exposure, antibiotic exposure. If the mother is taking during pregnancy too many antibiotics, that is not good for the infant's immune system or different organ development, because antibiotic can cause the dysbiosis of the microbiota, and if there is a dysbiosis of microbiota, then there is a imbalance of the absorption of the nutrients, and that in lack of nutrients can cause the crippled organ development, or it can hamper the organ development of the neonatals. And also there is another hypothesis you remember, that is the hygiene hypothesis. Hygiene hypothesis is not related to any hygiene, is not related to any cleanliness. This is a misnomer. Hygiene hypothesis is related to the how diversified microbiota helps into develop a good immune system, so that vaccine can provide good memory response. So if you do not have the good microbiome diversity, in that case your hygiene, based on the hygiene hypothesis, your vaccine efficacy will be down. The whatever the vaccine you will take after exposure of that particular pathogen, you might have the symptom, you might have the infection. But according to hygiene hypothesis, if you have the good microbiome diversity, that helps for good immune system development, which provides the good memory response. And these are the critical factors, like microbiota, metabolic factors, and environmental factors. When it comes into individual, followed by in your family, followed by in your society. In that way, it develops a herd immunity. Like you remember that in 80s onwards, polio vaccine has been given in pan India. So it was a mission-based project by the government of India, through which every all the in villages there was a polio vaccines were given whoever born, and after 30 40 years you can see that all the humans in pan India they develop a immunity against the polio virus. So we do not have much polio infected patients in our society. In that way we develop the society immunity or herd immunity. Similarly, we have generated the immunity against the SARS-CoV-2. We have generated the immunity against the leprosy, and this we generate the herd immunity or society immunity against a particular disease or pathogen. So when in your society there are three types of people you remember. You have the immunized people who has the vaccine. You have the less susceptible people who has the resistance capacity, and you have the sick people. That is also in your family also. You might see that same food you have taken, uh, one person in your family gets sick, another person is absolutely fine. So in your family, in your society, you can see that this type of three population you will find which are immunized, less susceptible, and which are highly susceptible. And this microbiome actually can alter this less susceptible or sick susceptible to highly resistant, or highly resistant person to less susceptible or severely susceptible based on the microbiome population. So if you have a good microbiota, this microbiota after having the good microbiota, if you have the infection, that provide, then you can see that immunized people are getting more. So slowly the most of the in the cohort, most of the people are getting immunized, and in that way you can develop a herd immunity. But if you do not have the good microbiome, there is a dysbiosis, followed by there is an infection, there is a immune dysregulation, and you can see the number of susceptible people are more, because people will vaccine efficiency, because vaccine is not working 100%, and in that way there is a sick or contagious people will be more in your society or in your community. So microbiome is the critical factor which plays an important role for immune system development as well as vaccine efficacy. So herd immunity or society immunity is the critical therapeutic intervention that delivers protection against infectious diseases both individually at the along with the community level. And this is entirely supportive while fighting against any disease in pandemic circumstances where enough people are susceptible to the infection. The conventional idea of herd immunity is established on the relationship between transmission dynamics of the infectious agents and natural population immunity. And this method is of natural vaccination. This is known as natural vaccination, is very much pertinent to the current COVID-19 when it was corona infection was there. So here you can see that this is the coronavirus. Perhaps you have seen, everybody knows it has the different proteins, and one protein is the spike protein, and spike protein is the critical protein. Based on that, all the vaccines are developed, either mRNA vaccine or DNA vaccine. All the vaccines mostly they were generated targeting the spike protein, and on the Covaxin, it is the whole virus which was attenuated. So Covaxin has that all the proteins, but other than Covaxin, Covishield, or any other vaccine against the SARS-CoV-2, they are mostly generated targeting this RBD portion or the total protein of the spike. So there is the alteration of the microbiota due to infection. We have shown that, and these are the. Now the question is that do we have time? How much time I have? or I'm
Yes,
done?
yes, you can continue. 20 minutes you can take. 15 to
Okay.
20 minutes.
So
Yes.
I will not take more than 10 minutes.
Yes, yes,
So,
please continue.
you have perhaps you have seen that many people have shown that the H2 androgen, this H2 is the receptor which actually present in our body, that is actually can form a interaction with the spike protein, that is the receptor binding domain of the spike, which makes a interaction, and in that way the SARS-CoV-2 can enter. But in our lab, we have seen that there are T cells, B cells, NK cells. They do not have the S2. But how and but in those cells, the virus was found. So now question is that if those cells they do not have the S2, how the infection happens in those cells? Here you can see that there are multiple papers who have shown that S2 independent infection in T lymphocytes, S2 independent infection in of SARS-CoV-2. So these are the question have been come in mind, that where H2 is not present, this H2 is not present, how the infection happens. Now that this we have found that this we have found one of the receptor in our body, that is dipeptidyl peptidase 4 DPP4 on CD26. This is the another receptor present in our body which is being recognized by the receptor binding domain of the spike. And here we have shown that this DPP4 can bind with the RBD of the spike protein. Here you can show the interaction, and this is the hydrogen hydrophobic interaction we have shown. I am not going in detail. And here you can see the interaction of the, this is the simulation data, and where you can see that there is a strong hydrogen and hydrophobic interaction is there. And after 100 molar of 100 nanoseconds of simulation, there is no dissociation between these this is between the RBD as along with DPP4. So here we have shown that this is the RBD which are expressing at the membrane, and this is the DPP4 which are expressing at the membrane, and you can see there is an interaction, means RBD and DPP4 can interact, and that's why you can see there is a merging of red and green. That's why it's showing the yellow. And we have done the immunoprecipitation of the DPP4 where RBD was expressed, and you found that there is a RBD DPP4 interaction. And then you know that DPP4 is the enzyme which is causing the diabetes too. But because DPP4, if it is more, it helps in suppression of insulin secretion. So there are multiple drugs are there, which is gliptin family along with metformin, which inhibits the DPP4 expression. Sitagliptin, linagliptin, sexagliptin, betagliptin, metformin. These are the different gliptin family drugs which inhibit the DPP4, and we have tried to inhibit the DPP4 RBD complex. And here we can see that due to using these drugs, there is a dissociation between the DPP4 RBD. And then what we have done, we have taken the live virus. This is the one virus, and we have used in different concentration of sitagliptin, linagliptin, sexagliptin, metformin, and we have found that sitagliptin is significantly working here. You can see that there is a significant virus load goes down. So here you can see that sitagliptin, linagliptin, or in combination can inhibit. This is the viral load in normal cell. But when we have given sitagliptin or linagliptin or in combination, you can see there is a significant inhibition of the multiplication of the SARS-CoV-2 virus. So sitagliptin, linagliptin demonstrated the greatest potential in terms of lowering the viral replication with IC50 of 1.46 micromolar and 2.2 micromolar respectively. And this is the simulation data, but we have shown that there is a dissociation of the RBD and DB4 due to sitagliptin as well as linagliptin. So here we have checked, this is the different RGR, RMSD or SASA value, and you can see that when we have given this is the normal one where you can see that DP4S2 can make interaction, but when we have given sitagliptin, niragliptin, or in combination, you do not find this yellow one. If you do not find the yellow one, means sitagliptin and S2, RB S2 and DP4 can express, but they do not make the interaction. So S2 and RBD cannot make the, DP4 cannot make the interaction here. RBD and DP4 cannot make the interaction here. Here you can see that RBD DP4 can make interaction, but when we have given sitagliptin, niragliptin, or in combination, the RBD and DP4 cannot make a complex. So you can, here we can tell that other than S2, there is another receptor which is DP4, where the spike protein, basically the RBD domain of the spike protein, can bind and virus can enter inside the cell. So here is the viability assay, and here these are the different, and along with the Wuhan virus, we have checked this efficacy of sitagliptin and niragliptin in different variants like alpha variant, beta variant, delta variant, kappa variant. And here you can see this is the alpha variant, where sitagliptin, niragliptin, or in combination can inhibit the virus growth. This is the another variant. This is the kappa variant. This is the delta variant, where you can see that live virus can be inhibited by using sitagliptin, niragliptin, or in their combination. So with that, I would like to. So this is the here you can see that this linagliptin, sitagliptin also inhibit the RBD-S2 interaction. Here you can see the RBD-S2 interaction, but linagliptin, sitagliptin also inhibit the RBD-S2 interaction, and that's why even there is a if you use this molecule, you can inhibit the DB4 expression as well as S2 expression, so virus cannot enter. So this is the model summary. Here you can show that strategy 1 is DPP4 RBD interaction. DPP4 RBD interaction can be inhibited by this drug. This is sitagliptin, niragliptin are used for the diabetic patient, and this drug can also inhibit the S2 RBD interaction. These are the different papers where we have shown that this is that another paper where we have shown that sorafenib, that is the cancer drug, which also can inhibit the SARS-CoV-2 RDP, that receptor, that protein, that enzyme which helps in RNA replication, that also inhibited by RNA-dependent RNA polymerase is being inhibited by sorafenib, and that we have published in PLOS Computational Biology. Here also we have shown that the DPP-RBD interaction is important for SARS-CoV-2 entry. Also, we have worked in monkeypox, where we have generated a novel vaccine using the reverse vaccine technology. And these are the different information where we have shown that why polio vaccinated people are more resistant. You remember that the kids were very much resistant to the infection. Why kids do not have the much COVID infection than older people? Because they have the COVID polio vaccine, and polio antigens has the, we have checked, they have the homology with the SARS-CoV-2 antigens, and that's why it provides the heterologous immunity, and heterologous immunity helps to provide protection to some extent against the SARS-CoV-2 infection. And this is my lab, and these are my collaborators, and these are the different supports we have from the different agencies. Along with that, I would like to stop here. Thank you.